So over the next few moments, I'm going to be sharing with you some of the research that we've been doing in our lab, looking at ways of accelerating the translation of intranasal oxytocin treatment by improving methods, theory, and reproducibility. Now, when it comes to oxytocin, something which is often associated with oxytocin is hype. When it was first tested within animal models, it's almost quite a long time ago. It was found to be associated with social behavior in animals. Then there were a few studies in humans in the 80s, but things didn't really take off until a study came out in 2005, which reported that oxytocin increases trusting behaviors. And this set off a whole bunch of studies looking at oxytocin's role in social behavior. Of course, this is of particular interest for psychiatric disorders which are characterized by social dysfunction, such as autism and schizophrenia, for instance. So, as well as looking at oxytocin's role in social behavior in neurotypical individuals, researchers began to investigate and report some very exciting findings looking at oxytocin's effects on manipulating social cognition and social behavior, such as improving theory of mind performance, but also increasing gaze to the eye region. So there was a lot of excitement and a lot of promise for what oxytocin could do, because then, and still to this day, there is no pharmacological treatment specifically targeted to improving social behavior. So this particular treatment, which is an old treatment, which has been around for a number of decades, but potentially could have been repurposed for addressing social dysfunction, held a lot of promise. And there was a lot of excitement, which led to a lot of panacea claims that oxytocin could be used to treat a whole bunch of things, including anxiety disorders, depressive disorders, a whole range of different disorders across the spectrum of psychiatric illnesses. And of course, this also led to a number of hyperbolic articles in the media proclaiming oxytocin to be some sort of wonder drug. So there was a whole bunch of visibility and a whole bunch of expectation around oxytocin. But as you're probably aware, things came crashing down when a number of studies had trouble replicating some of these initial results. There are troubles replicating the trust studies, but also oxytocin's effect on social functioning. And so people began to get quite disappointed because the original expectations weren't actually matching reality. And then there was a few high-profile cases of oxytocin having no significant effects for autism or for schizophrenia. But then we reached this so-called enlightenment phase where we began to get a better understanding of oxytocin's effects, that it wasn't just a hormone that influenced all areas of social cognition and didn't just automatically treat whatever disorder that it was administered to or the population that it was administered to. But people began to understand that oxytocin was a bit more nuanced, that the individual and the context matters. And it also depends on administration and experimental methods. And once researchers began to get a better understanding of the importance of context and individual, some more positive results began to emerge. So oxytocin has been on this roller coaster of hype, and I believe we're still within this so-called enlightenment stage, but there still is a long way to go in order to reach this so-called plateau of productivity. We need a better understanding of what oxytocin does, and that is where a lot of my research steps in. How can we accelerate this translation of oxytocin to this plateau of productivity? We published this paper, I think about 2 years now, which outlined ways to improve the precision of intranasal oxytocin research, which includes a lot of work from our group, but a lot of work elsewhere too. And part of the major argument for this particular paper, which was led by a former PhD student, Adriano Winterton, was that in order to accelerate the translation of intranasal oxytocin research, researchers need to address three different things: methods, reproducibility, and theory. Now, perhaps if you were just to address theory or improve theory and methods, you may have a good research question and a well-conducted study, but the results may not be reliable. If you were to just address methods and reproducibility, you may have a reliable effect with good methods, but you might be asking the wrong question. And if you were just to address theory and reproducibility, you may have a good research question with reliable effects, but inefficient methods. So you need to address all these three things. And there's been for quite some time concerns around methods and also concerns about theory and reproducibility, but very seldom were all these three concerns addressed. When it comes to methods, an important consideration which often isn't included within research studies is the importance of auxiliary hypotheses or auxiliary assumptions. When you have a set of hypotheses, these are always paired with an auxiliary hypothesis. So this is from a paper that was published last year in Psychoneuroendocrinology. To use the experimental model as an example, you may have an hypothesis that intranasal oxytocin administration increases behavioral flexibility, but this relies on the experimental auxiliary hypothesis that intranasal oxytocin administration actually increases oxytocin in the central nervous system. Now, the reason that this is critical is that this is required in order to falsify hypotheses. If you do not have strong auxiliary assumptions, it is very difficult to falsify hypotheses, because if you get a non-significant result, then it's quite easy to blame the auxiliary hypotheses. You could say, well, we did get an effect, but then again, we're not entirely sure whether oxytocin actually reaches the brain. Or you might say, well, we're not entirely sure whether we use the correct dosage. It's critical to address these auxiliary hypotheses in order to give to have the opportunity to actually falsify hypotheses. If we cannot falsify hypotheses, we cannot move theory forward, and we'll continue ahead. And this is and not getting a better understanding of what's happening because we cannot falsify these hypotheses. Something which I touched on with that example before is this very question: does intranasally administered oxytocin reach the brain via a direct nasal route? And what is the most effective dose? These are really important questions for oxytocin research. Traditionally, oxytocin is administered via the nose because this offers a direct route through the nasal cavity, transported through the trigeminal and olfactory nerve fibers into the brain. At least that's the assumption. Of course, you could administer oxytocin intravenously, and this is what's done quite commonly in childbirth, but because oxytocin is a relatively large molecule, it has difficulties crossing the blood-brain barrier, because our goal is to increase levels of oxytocin in the brain to activate oxytocin receptors. And that is the idea behind intranasal oxytocin administration, that you can bypass the blood-brain barrier and administer oxytocin traveling down these olfactory and trigeminal nerve fibers. So that's one assumption, and the other assumption is that we're actually using the correct dose. So a number of years ago, I began a series of trials answering these very questions, which helps when it comes to addressing these auxiliary hypotheses. So these first sets of experiments ask the question: what is the impact of dose and the peripheral effects on cognition, neural activity, and physiology? For this particular experiment, we had 4 different conditions using a double dummy design, in that we used 2 types of administration routes: the intranasal route and also the intravenous route, because we wanted to compare the effects of oxytocin administered intranasally and oxytocin administered intravenously. So by doing this and comparing the effects of these different modes of administration on cognition, neural activity, and physiology, this would provide evidence, or this would provide the opportunity to examine what is more effective. Is it intranasal or is it intravenous? If there was effectiveness after intravenous oxytocin, and there's certainly been work to demonstrate that that's the case, then this would suggest either two things. One, that intravenous, that circulating oxytocin administered intravenously does indeed cross the blood-brain barrier, or it would suggest that the activation of peripheral receptors, and the body is full of oxytocin receptors outside the brain, would have is having some sort of feedback effect to the central oxytocin system. But regardless, this is a good way of examining is the nasal route a privileged route. The other implication would be that perhaps the way that the intranasal oxytocin works is that it simply increases oxytocin in the blood. We do know that intranasal oxytocin administration increases oxytocin levels in the blood. So rather than actually entering the brain via a nose-to-brain route, it simply increases oxytocin in the blood and crosses the blood-brain barrier that way, or activates peripheral oxytocin receptors. So that's why we had this double dummy design. In the first condition, we looked at a lower dose of oxytocin, 8 international units, or 8 IU, and compared this with a placebo IV. In the second condition, we used a higher dose, 24 IU, which is the most common dose that's used in oxytocin research. And there isn't actually a very strong justification for why this particular dose is used, other than this is what the last person did. We don't actually know what the most effective dose is. So that's why it's important to do these dose comparison studies. Condition 3 was placebo intranasal oxytocin administration and oxytocin intravenous. And the fourth condition was placebo and placebo. The other thing I want to mention as well is you're probably looking at these nasal spray devices thinking that's not what a nasal spray device looks like. And that's true. We worked with a company called OptiNose who has developed technology to improve nose-to-brain delivery of nasal sprays. Traditional nasal spray devices were never designed to deliver drugs to the brain. They're only designed to deliver drugs to the nose and into the bloodstream. These particular devices are designed to administer nasal sprays to the upper and posterior regions of the nose, which will increase the ability for administered drugs, including oxytocin, to reach the brain. So that's why these nasal spray devices look a bit different than what you're used to. So we have these 4 different conditions. And the first thing we wanted to look at was, well, what is the effect of circulating oxytocin as measured in the blood? And as you would expect, there was quite a high jump of oxytocin concentrations after intravenous, which came down pretty quickly, which reflects the fact that oxytocin, at least in the blood, tends to be broken down quite quickly. But then when you actually compare this, at least after the 10-minute mark, the levels of oxytocin in circulating blood was comparable between the IV administration or the intravenous administration and the 2 doses of intranasal oxytocin. And this is important because this suggests that regardless of the administration route, you tend to get the same amounts of circulating oxytocin in the blood, at least. And then we administered a social cognition task in an MRI scanner about 40 minutes after oxytocin administration. So what did we find? Well, we actually found that a lower dose seemed to be more effective in modulating social cognition or social cognitive behaviors in a neurotypical population of young men. And compared to placebo, it was the smaller dose, the 8 international units, and not the larger dose, nor the intravenous dose. We also looked at a very common target of oxytocin and fMRI studies is the amygdala, because there's been many reports of oxytocin reducing amygdalar activity, at least within males. So we found the same results in that compared to placebo, only the lower dose significantly reduced amygdalar activation. And because we only found this effect through the intranasal route, despite the fact that we had comparable concentration levels after intravenous and intranasal, this suggests, this at least is consistent with the idea that intranasal oxytocin reaches the brain via a nose-to-brain route and not via the circulatory system. And it also suggests that a lower dose might be more efficacious. And it's not just us. Subsequent work has also demonstrated that a lower dose might be more effective as well. So from Daniel Martins and co-workers have come to similar types of conclusions. And the same group has also found that the effects of administration also supports a nose to brain route as well, at least for specific brain regions too. The next question is how we can improve the reproducibility and reliability of intranasal oxytocin research. The psychological sciences and the medical sciences have been undergoing a reproducibility crisis where a lot of studies have failed to reproduce. And as I mentioned before at the start of this talk, oxytocin is no stranger to this. So it is important to think of ways of increasing reproducibility. Of course, when it comes to hypothesis-driven studies, pre-registration is a very important factor of increasing reproducibility as well. And there are various other initiatives such as the Registered Reports initiative as well, which is another way of pre-registering your studies. One important factor for reproducibility is statistical power. There's a highly cited study which came out a number of years ago that suggested that most intranasal oxytocin research is statistically underpowered, which means that they cannot reliably detect a range of effect sizes. A couple of years later, I redid this again, including more recent studies, and unfortunately, at least when this was done in 2020, I think it was including studies up to 2019, there hadn't been a big improvement, and this is critical if we are to increase the reliability of oxytocin research, and it's critical for any research field. So this particular study was used using the meta R package, which is something that I've developed, and there's a preprint available for that particular study, for that particular R package, to do these analyses and to create these fire plot figures, which demonstrate the range of effect sizes that can be reliably detected. And for all these meta-analyses, this looks at the median the median statistical power for a range of hypothetical effect sizes. And for most of these analyses, the range that can be reliably detected, and typically this is 80% power, there's quite a range there. So although for some subfields it's improved, for others it hasn't improved so much. It is very common to think about statistical power once a positive study is published. Quite often you'll see there's a positive study, and one of the first questions that's asked is, well, the sample size is quite small and the study is underpowered, therefore we should, it's unclear how credible the study is. But this also works the other way, in that you need appropriate statistical power when it comes to reporting and thinking about non-significant studies. Most non-significant studies within oxytocin research have been underpowered to also reliably reject a wide range of effect sizes. So in this particular study, which was published 2 years ago in Comprehensive Psychoneuroendocrinology, I looked at a number of studies or a number of meta-analyses, some of which had non-significant results and concluded that yes, there's no effect here. But assuming that the effects of intranasal oxytocin administration are smaller than 0.2, which is quite a fair assumption, only 4 out of these 26 meta-analyses demonstrated statistical equivalence. So essentially, the sample sizes were not big enough to reject to reject a realistic range of effect sizes. And if we were to assume that Cohen's d of 0.1 is too small to care about, no meta-analyses demonstrated statistical equivalence. So the implication here is that we need appropriate statistical power, both for significant studies, but also for non-significant studies as well. One element that's been neglected in terms of reproducibility is the importance of theory. It's been suggested that one of the reasons we're not actually getting reproducible results is that we're actually asking the wrong questions in the first place. So this really speaks to the importance of improving theory to ask better questions. This is Nico Tinbergen, who co-won the Nobel Prize in the late 70s for his work on social behavior in animals. And one of his more well-known approaches for understanding how phenotypes works was by asking 4 questions. How did this phenotype evolve? How did this phenotype help survival? What is the physiological cause, as in the mechanism? And how does this phenotype develop in the individual? So in terms of better understanding oxytocin research, I've been using this approach for better understanding what oxytocin does and for improving theory. Here is some new data, which will be soon published as a preprint, which is an investigation of how the oxytocin signaling system evolves. Quite often oxytocin, when it comes to genetics, is focused on 3 genes: the oxytocin receptor gene, CD38, which regulates oxytocin secretion, and the structural gene for oxytocin, OXT. Yet there is a large signaling pathway which contributes to oxytocin, which contains 154 genes. So what my PhD student Alina Sartorius did was she calculated the gene ages of all the genes in the oxytocin pathway using BLAST-P and microsynteny across 41 species spanning all major evolutionary lineage splits. People have looked at oxytocin genes at a few species, but this is the first systematic look across 41 different species. And by doing this, we were able to determine the ages of all these genes here. So we hope to release this preprint within the next few weeks, so keep an eye out for that. And one thing that we found was that more modern oxytocin pathway genes, or genes that evolved more recently, are enriched in muscle and brain tissue, whereas more ancient genes seem to be enriched in blood vessels and the bladder, or more constricting organs. So that's exciting work, and I'm looking forward to the preprint getting published there. Now, another approach for understanding what oxytocin does, or the purpose of oxytocin, is by looking at oxytocin receptor expression patterns. And there's been a lot of work looking in animal models of oxytocin receptor expression. And one particular study a particular line of work has manipulated oxytocin receptor expression by both increasing it and decreasing it. And this has demonstrated that you can manipulate social behavior by simply manipulating oxytocin receptor patterns in the brain. So this suggests by adjusting these patterns, all these patterns have an important role to play in particular behaviors. But to date, there hasn't been much work looking at oxytocin receptor expression in the human brain. This is very labor intensive, and traditionally only 2 or 3 sites have been measured. Yet now, but by doing this, we can actually look at which mental states are related to oxytocin receptor expression patterns in humans. So if, for instance, we were to look at oxytocin receptor expression patterns across the brain, then we could associate this with known regions that are associated with different mental states. When you're looking at oxytocin receptor expression patterns in specific regions, that doesn't give us that much information, because mental states are supported by neural networks. So by looking at the entire brain, we can do this. And fortunately, we're able to do this using the Allen human brain dataset, which samples gene expression as measured by mRNA across hundreds of samples across 6 different brains. And by doing this, we could do this and get a gene expression for over 20,000 protein coding genes, and then create voxel by voxel expression maps for all 20,000 protein coding genes, including the oxytocin receptor. Our first bit of analysis found that the oxytocin receptor gene was enriched in subcortical and olfactory regions in this particular sample. And also the olfactory region, which is very interesting, considering the other work that we've been doing, suggesting that oxytocin reaches the brain via the olfactory nerves, which first enter the brain near the olfactory bulbs. So this is interesting in that context. So by doing this, we're able to create a voxel by voxel map of oxytocin signaling genes to see exactly where in the brain these genes of interest, particularly the oxytocin receptor, are located. But then what we're able to do is to figure out what mental states these were created with. We combine this with a large-scale fMRI meta-analysis of over 14,000 studies. So, for instance, using this meta-analysis, you could have a particular mental state such as pain, and then this would search this corpus of studies, and any studies looking at pain, then it automatically extracts the coordinates, and you're able to create a map of neural activation related to the processing of pain. So this is one way of doing it. It was so-called forward inference. You have a mental state, and you would actually see what areas of the brain this is related to. But what's more powerful, but by doing this, it could be related to a number of different mental states. What's more powerful is doing reverse inference, in that you actually provide a brain activation map, and then you see which mental states of the entire corpus that these are most strongly related to. And this is precisely what we did for our particular research. We took these oxytocin receptor maps to see which mental states, out of the hundreds of mental states within that were reported within this corpus of studies, were most strongly related to the oxytocin receptor gene. And we found the oxytocin receptor expression pattern was associated with anticipatory, appetitive, and aversive mental states. And this was also specific to the oxytocin receptor gene, and also these two other important genes in the oxytocin pathway. So what we did was we took these particular mental states, and then we calculated the correlation with not only these genes of interest, but all 20,000 protein coding genes, to see was the oxytocin receptor among the highest correlations for all genes for these particular mental states. And for things like reward, and for things like incentive or anxiety, there was a number of different hits where the oxytocin receptor was among the top 0.5% of relationships compared to other genes as well. So this demonstrates specificity. We also did an investigation saying, well, what other genes is the oxytocin receptor most highly related to? And the highest hits were linked to metabolic regulation, and the most negative hits or negative correlations were genes linked with schizophrenia. So there were some really interesting outcomes here. In terms of the purpose of the oxytocin system, another thing that we looked at, or we've been looking at, is the link between psychiatric illness and metabolic dysfunction. Looking at schizophrenia, for instance, schizophrenia not only is associated with social dysfunction, but also with metabolic dysfunction over and above the effects of antipsychotic medications. And a number of years ago, we suggested that perhaps oxytocin system dysfunction could be a common link between these two things. So to investigate this, we created an oxytocin pathway polygenic score using schizophrenia GWAS. And then we associated this polygenic score with metabolic variables in the UK Biobank, which has just under 500,000 participants. And we also accounted for important covariates as well. And then to test for the specificity of these effects, we created polygenic scores for 100 random gene sets of the same size. So although we may have found a significant association between the oxytocin pathway polygenic score and these particular factors, we created these other gene sets to see whether there was specificity for these effects. And we did find significant and specific relationships for the oxytocin polygenic scores and various metabolic variables, as reported in this paper, which was led by Adriano. And more recently, we looked at the association between loneliness and metabolic health. And to do that, we created an oxytocin pathway polygenic score for loneliness using GWAS data. And then from this, we linked this to body composition and metabolic variables in the UK Biobank, again, accounting for important covariates. And we did indeed find specific effects for this loneliness polygenic score and metabolic variables as well. And this link was also found from when we created an oxytocin polygenic score for BMI, and then linked that with other various body composition scores as well, which were calculated using body MRI, but also direct measures of bone density as well. So some interesting links between the oxytocin system and loneliness and cardiometabolic health. Another important perspective in terms of development is oxytocin's role across the lifespan. Oxytocin seems to be heavily investigated in young adults because that's how old undergraduates are. But there needs to be a stronger push for actually looking at where oxytocin across the lifespan, which we mentioned in this opinion piece, which we published earlier this year. And to this end, we took the same approach for our gene expression and looked at oxytocin receptor expression across the lifespan. Remember, before this original study that I showed you, this was done in adults. I believe the average age was about 50. Whereas for this particular study, we used 57 male and female donors from the first trimester all the way to adulthood to see what periods of life are associated with increased oxytocin receptor expression pattern in people who are otherwise healthy. And we found that the highest peak was during early childhood. And once you actually split this between males and females, it seems to be stronger in males as well. So this is a really exciting study in that we're able to pinpoint exactly when during development oxytocin receptor expression is most increased. So all that to say, it seems there's more and more evidence that oxytocin's effects are both social and non-social as well. Researchers traditionally focus on oxytocin's social role, but accumulating evidence suggests that it also plays a non-social role as well. And all these things together, this research has led to the proposal of this allostatic theory of oxytocin, that oxytocin helps regulate stability in changing environments by influencing these four factors, by sensing changes in the environment, by influencing learning, the prediction of future events, and responses to changes in this environment. So this is an effort to improve theory based on the evidence using Tinbergen's 4 questions. And this is something that I'm going to continue investigating. And of course, this theory is open to be adapted. Theories that aren't necessarily concrete and should be adapted. So I'm going to continue investigating these different things to see if there are ways to improve this theory that we've proposed. Again, like I mentioned earlier, this is absolutely a team effort. So I want to give a special thanks to past and current PhD students and team members who have contributed to all this work. I'm looking forward to answering your questions. Please use the app as well, or you can send me an email if you have any questions as well. But I thank you for your attention.